Now that we have that out of the way, what are your questions? Yes, sir.
Okay, to clarify, in blue white ribbon, when the lac Z gene is present,
Yes.
it produces blue color, correct?
So when the lac Z, that's the thing. To clarify and make sure we're absolutely clear, when in the lac Z system, okay, that we used for plasmids that I was talking about, when beta-galactosidase is present, that's lac Z, okay? When beta-galactosidase is present, it cuts XGAL and produces a blue color. If beta-galactosidase is not present, it will leave the material white. Yeah, I screw that up every time I do it. I don't know why. I have a mental block about it for some reason.
So starting off with white.
You're starting off with a plate that has no color. So, and the cells are white when they have no beta-galactosidase. Yeah. Blue is confirmation. No, it's not what you want. Blue is confirmation that you have beta-galactosidase present. Remember, you want to get an insert. So the insert destroys the beta-galactosidase. So you're looking for white. You're looking for white color. Those will have inserts. White colonies will be created wherever you have an insert into the beta-galactosidase gene. That's what you're trying to determine. Do I have an insert? I want to make sure I get an insert, because most of them are just going to come back together and remake beta-galactosidase. Okay? Beta-galactosidase is the lac Z gene. That's correct. That's fine. Sure. Let me get Anna here first.
Can you explain
Microarrays, sure. So let's go back to biotechnology, outline, microarrays. All right, microarrays. So what are we trying to do in microarrays? Well, there's a variety of things that we can try to do. One I talked about in class is doing what's called transcriptomics. So we're trying to see what genes are being transcribed. That's our aim. Okay? How do I do it? Well, I know the sequence of every gene. So I go to my friendly chemist and say, sequence the complementary sequence to every gene. I'm going to take gene number 1, I'm going to put all those on this spot. Gene number 2, put them all on this spot. Gene number 3, put all of them right there on that spot, etc. And I keep track of which ones I put where. The complement to the messenger RNA. That's right. You with me? So spot number 1 might have DNA polymerase. Spot number 2 might have a ribosomal protein. Spot number 3 might have beta-galactosidase, etc. I keep track of which spot is which one. Okay? Then I take my cells and I bust them open. And I isolate all the messenger RNA out of those cells. And I label them with a color. Let's say I put green on them. Okay? Put green on them. If I just did green, what would I have? When I put them on here, I would be able to tell wherever there's green, I would know that gene was being made because I'm letting them hybridize so they find their complementary sequence. I wash off the unhybridized stuff. Anything that's green is being made. The amount of green is going to be a function of how much of that gene is being made. And if I have no green, I know that gene is not being made. Now, this is a more complicated one because I've got 2 types of cells. Messenger RNAs from one cell I label green, and the messenger RNAs from a different cell I label red. Let's say one is a normal cell, the other one is a cancer cell. Red in the cancer cell, green in the normal cell. I take those messenger RNAs and I mix them up. And then I pour them on the plate and I let them hybridize to find their complementary sequence, and I wash away the unhybridized stuff. Now, red plus green equals yellow. Wherever I see yellow, it means both of them are being made in equal amounts. Wherever I see red, it's being made only in the cancer cell and not in the normal cell. Wherever I see green, It's being made only in the normal cell and not in the cancer cell. The intensity of each of these is a function of how much of each one is being made. So if I see intense yellow, like I see here, they're both being made abundantly and in equal amounts. If I see a faint yellow, let's see, I'll find a faint yellow on here somewhere, but the faint yellow right here, I would say they're not being made very much in each cell, but they're being made in equal amounts. Intense green, a lot in one versus the other. Light green, a little bit in one, in the normal versus the cancer, right? Same thing with red. So the combination of all those tell me which genes are being transcribed inside of the respective cells. Make sense? Okay, yes?
Would this change if you took examples at like different times with different proteins?
Sure. So our question is, would this change if I did them at different times? I could actually do this. I could take one batch of cells and collect them at 5 minutes after doing something to them, and another one at 20 minutes after doing something to them. And I could say, in that time frame, did something change inside those cells? And I could answer that question real quickly. Yeah, sure could. I can do any kind of treatment I want. I can say, give me this one a drug, don't give this one a drug, right? Or I could say, let this one be old cells that have been growing for many generations, and this one be brand new cells that just got made by fertilization, for example. Okay, other questions? Yes? Oh, the one that we called TRNA enzyme, right? Which I thought was a dumb name, but that was just me. So, okay, what's the function of tRNA enzyme? The actual name of tRNA enzyme you don't need to know, but it's the amino acyl tRNA synthetase. And that molecule, if I can pull up my stuff here, that molecule is right here. Okay? What is the amino acyl tRNA synthetase? What does the tRNA enzyme do? Something very simple. It takes a tRNA, it reads the anticodon, and it knows which amino acid to attach to the other end. And it does that. That's all the tRNA enzyme does. It reads the anticodon, makes sure it's got the right amino acid to put on the other end, and that's that. There's one tRNA enzyme for each amino acid. Yes, it's the 3' end of the molecule. So in that cloverleaf structure, that longer end at the very top is the 3' end of the molecule. Question?
So does synthetase mean that the two aforementioned molecules are being joined?
Yes. Yes. So the amino acid is being covalently linked to the tRNA. And remember, it's that joining that is very unstable in water, which means you got to have EFTU real close by to cover it up real quickly. Otherwise, it's gonna fall apart.
Synthase, you could call it, I guess, this is just main convention thing, but if you say something is synthase, that means that it makes the molecule that it's the name.
Okay. So people used to distinguish synthase from synthetase, but the two terms are used interchangeably now. So that was a distinction they did away with some time back. Yeah. So synthase equals synthetase. Yes, ma'am.
Is there anything that brings the amino acids to the specific tRNA, or does it just come together based off like
Okay, so is there anything that brings the amino acid to the synthetase? The synthetase itself will bind to the amino acid.
that allows the tRNA to come?
Yep. So it's bound to the enzyme,
And that's the TRNA enzyme.
the tRNA enzyme.
The tRNAs themselves can have the I, the wobble base on it.
The tRNAs can have in their codon the wobble base in, I'm sorry, the tRNAs will have in their anticodon, I'm saying it wrong myself, the wobble base in the 5' end of the anticodon, right? Don't confuse the anticodon with the codon. The codon is found in the messenger RNA. The anticodon is found in the tRNA, and they're complementary to each other. Yes? 5 things to make a protein? Okay, well, the things that we need to make a protein will include a messenger RNA, a small subunit ribosome, a large subunit ribosome. You'll need the appropriate tRNAs, and you will need energy, which would be in the form of GTP. So if there's 5 things, I guess that's what, I can't remember saying 5 things, but if I said 5 things, those would be the 5 things. Okay? Yeah, good question. So do prokaryotes and eukaryotes both have initiation factors? The answer is yes, they do. Everything in eukaryotes that I didn't talk about is the same as it is in prokaryotes. They just have different names, and so rather than confuse you with a bunch of names, I left that out. But they have the same things. They have initiation factors, they have elongation factors, and they all have termination factors as well. John. Of the ribosome? Yep. Yes. So the question was, the small subunit of the prokaryotic ribosome, the 30S subunit, contains the 60S ribosomal RNA. Yes, that's correct. And is it the 16S ribosomal RNA that contains the sequence that's complementary to the Shine-Doe-Garneau sequence in the messenger RNA? And the answer is yes, it is. Yes, sir. Yeah, I've had a couple people ask me that question. Is there a difference between a plasmid and an expression vector? A plasmid can be an expression vector. Okay? So the term plasmid just simply means a circular DNA replicating in E. coli. So it becomes an expression vector if we put a promoter in that plasmid. But a plasmid per se doesn't have to have that. So a plasmid is a big term. You know, an expression vector would be a subset of plasmids. We use plasmids to do a lot of things. Mariah? What does the 23S ribosomal RNA, it's not a subunit, it's the 23S ribosomal RNA, in the 50S ribosomal subunit do? Okay? The 23S ribosomal RNA catalyzes the formation of the peptide bond during the process of translation. Catalyzes the formation of the peptide bond during the process of translation. Yes? I didn't say a function for the 5S. It's not completely understood, but it's thought that 5S might be involved in the translocation process of moving the ribosome along the messenger RNA, but it's not completely known. Yes? Can I explain the role of glycoproteins? Well, glycoproteins have many functions. The only function of glycoproteins that I've talked about in class is cellular identity. So cellular identity occurs when glycoproteins have specific carbohydrates on their surface, and that sticks out of the cell. So we could think of this as like an address. Okay? Your house has a number on the outside that says this is the house number for this. A cell could have the same thing. That could say I'm a liver cell. It could say I'm Kevin Ahern's cell. It could say I'm a blood cell. Okay? A variety of things that it could say. And your body will ignore those. That's good. That is, your body will ignore those things because if it recognizes them, it means the immune system is going to attack them. So you don't want your immune system attacking your blood cells, your liver cells, your brain cells, because if you have that happen, you have what's called an autoimmune disorder. When people have lupus, for example, their immune system is recognizing and attacking a part of their body. So as long as your body's ignoring all those signs, you're fine. If you get an organ transplant, or if you get a wrong blood type put into you, both of those, the immune system says, invader, attack, attack, and then you're in trouble. Okay? But that's done by the immune system recognizing that carbohydrate that's on the glycoprotein on the surface of cells. There are other functions of glycoproteins, but that's the one in class that I've mentioned. So the glycoprotein, let's listen to the name here. What does the name tell you, John? Glyco meaning? What does protein mean? Okay, so it's sugar attached to protein, right? This is a lot easier if you listen to the words, right? Glycoprotein is a sugar attached to a protein. Yes, ma'am.
Why is selenocysteine,
Selenocysteine,
not considered post-translational?
Why is selenocysteine not considered a post-translational modification of a protein? That's because it's actually, the selenocysteine is actually made in the protein during the process of translation. That is, it's attached to a tRNA, and during the process of translation, it gets built into the protein. For it to be a post-translational modification, it would mean that something had to happen to the protein after it was made to put the selenium in there. And that's not how the selenium gets there. Okay? Selenocysteine. Yes, sir. So the innate immune system is something that we walk around with that gives us general protection against a variety of invaders, most commonly bacteria. So there are certain common things that are found in the cell walls of many bacteria. And our innate immune system has some proteins that will bind to those and basically disable them. So the innate immune system gives us sort of a general protection against a wide variety of organisms. The adaptive is, as it says, adaptive and specific. That's correct. That's one way of looking at it, yes. Mm-hmm. Yes, ma'am.
The long-terminal repeat is a promoter on the double-stranded viral DNA.
Yes. So the question has to do with the retroviruses that I talked about, the long-terminal repeat. And her question was, is that something that's on the end of the viral double-stranded DNA? And the answer is yes.
And it got there by being reverse transcribed on the...
Right. So the viral double-stranded DNA came from reverse transcription of the viral RNA. That's correct. Yes, sir. Oh, going through the viral life cycle, is that what you're asking? Make sure I'm answering your question here. So you want... Uh-huh. 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 Is that what you want me to go through? Okay. Okay. So question is, if I have a single-stranded retroviral RNA, what are the steps in the overall infection process of that viral retroviral RNA? Okay, so first thing, for any virus anywhere, the first step is, of course, injection into the cell. That requires attachment. In the case of HIV, which I'm not sure if I said in class, I'll say it here. If I didn't say it in class, you're not responsible. The HIV actually binds to a CD4 protein that's on the surface of immune cells. That allows it to get into the cell. It injects its viral RNA and the proteins that it carries into the cell. The second step in the process is generally some sort of a replication and or translation of the genetic material that's there. In the case of HIV, the second step is actually the conversion of the viral RNA into double-stranded DNA. That happens as a result of the action of the reverse transcriptase that is in the cell. It does vary across viruses. Some viruses will start replicating before they do anything. Some will start translating before they do anything. So there's not a set order to all these things. Okay? But in the case of HIV, that's what's happening. So the viral DNA gets made in double-stranded form, and then the viral integrase protein integrates that double-stranded DNA into the cell's double-stranded DNA. Then it becomes a part of the cell. And that's the problem, as I said, with HIV, is once it's a part of the cell, not any good ways of getting it out anyway. All right? So whenever that cell replicates, that cell is going to replicate the viral DNA as well. And since that viral DNA contains those long terminal repeats that Anna asked about, that means that it's transcriptionally active, usually. And so transcriptionally active means it's going to start making viral RNA. Copy, 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 copy. And then that viral RNA can be translated to make viral proteins, to make reverse transcriptase, to make integrase, and then they can all be packaged up together into a viral particle and go infect. The last step in the process, of course, is making that particle and then releasing it out of the cell. Okay?
Reverse transcriptase takes single-stranded RNA, and does it make single-strand DNA first,
It makes single-stranded DNA first.
and then regular DNA-dependent DNA polymerase fills in the rest?
Okay, so it's a good question. It turns out the reverse transcriptase can make both strands. And I didn't say it in class, but HIV uses, you might think, well, how does it start making the second strand? You have to have a primer, right? You know what HIV uses to prime the second strand? It uses the end of a tRNA. Yeah, the end of a tRNA. I mean, these things take advantage of every possible thing that's there. That's what it uses to prime the second strand DNA synthesis. Right? Uh-huh. So the question had to do with plasmids and the efficiency of putting them into a cell. And I said in the highlights that at best you can get 1 in 1,000 into a cell. Meaning that if you took 1,000 cells and you threw all the plasmids in the world at them, only 1 of those 1,000 cells is going to take it up. Okay? So you want to have a way of identifying which cells take it up. And the way that we identify that is by the use of plasmids that contain antibiotic resistance. Because if we take our transformation mixture and we treat it with an antibiotic, only those that got plasmid are going to survive. So the 999 that didn't get it will disappear. They'll die. Yes? Can I explain how endonucleases and methylases protect against viral infection of bacterial cells? That's the question, I think, right? Okay, so let's think first of all about the endonucleases, which we also call restriction enzymes. We more commonly call restriction enzymes. Restriction enzymes recognize a specific sequence, bind to it, and cut it. That's what they do. Okay? So I gave you an example of the enzyme EcoR1, which recognizes the sequence GAATTC, binds to that, and cuts it. So if a virus is injecting a double-stranded DNA into the cell, the restriction enzyme is going to look for GAATTC. If it sees it, it binds it, it cuts it. When it cuts the virus can't function because that's necessary for replication of the virus. Okay? So I've protected the cell against the invading virus. The question then is, well, why doesn't the restriction enzyme cut the cellular DNA? And that's what comes from the methylase. So all systems that have all bacteria that have a restriction enzyme also have an equivalent methylase that recognizes the same sequence as the restriction enzyme, but instead of cutting it, it puts a methyl group on part of it. And that methyl group prevents the restriction enzyme from cutting. Yes, Mariah?
Protection against itself, like is the methylase protection against the bacterial restriction enzyme?
Yes. So the methylase is protecting against the restriction enzyme cutting it. So if the methylase has gone and methylated all the different sites in the cell's genome, the restriction enzyme can't cut the cell's genome because it's protection against that. All right? Now, the injected DNA from the virus hasn't had a chance to be methylated. That's how it gets cut. John, was there a question? I anticipated your question. Anna?
What is the implication of the overhangs?
What's the implication of the overhangs? Let me show you the overhangs without trying to describe them. So the overhangs for restriction enzymes only have a fairly minor significance, but I will still show you here. Where's my overhang? Here's an overhang. Okay, so EcoR1 cuts, and you see it's a what I called a symmetrical or palindromic sequence. GAATTC on that strand, GAATTC on that strand. It cuts between the G and the A on each strand. That leaves overhangs. Okay, and her question is, what's the significance of the overhangs? Well, it turns out these overhangs, if I cut another sequence with EcoR1, they'll have the same overhangs. So I have an overhang that goes AATT like that. Those base pairs actually help to stabilize it. So when I go to put the ligase in there, it's held together by those base pairs. That helps me in the laboratory to make recombinant DNAs. Okay? Helps me to make recombinant DNAs. Yes, question?
So in regards to EFG, I have it noted here that the amino acid moved from P site instead of P to A.
Okay. So, question had to do in the process of in the process of translation, what EFG was doing and the movement of things across the sites. So let's talk about that first. Where am I at here? Okay. So here's the process of translation. We are starting here. Okay. We've just finished the initiation process. We have a tRNA in the P site. That's the F-met, the very first one. We have the A site open. We bring in, okay, a second tRNA that's now complementary to this region here. So we got two tRNAs in the ribosome, as you can see here. The 23S catalyzes the joining of those. That causes the F-met to leave the first one and get joined to the alanine on the second one, like you see here. And now, after that has happened, we've done the reaction that we need. So now EFG comes into play, and we need to move the ribosome down one more. Well, what happens? EFG, using the energy of GTP, moves the ribosome down one codon. We can see that's happened here. We see that the initiation tRNA that was in the was in the P site, now moves over to the E site. The dipeptide that was in the A site has now moved to the P site, and the A site is again now open. Okay? Yes, sir. So let me think about that. So it's a little bit more complicated than that because the initiation factors use some GTPs in starting the process. But once you get going, the only GTPs that are involved are the GTP that's on EFTU and the GTP that's on EFG. So once you get started, yes, it's 2 GTPs per amino acid. Do protein-breaking enzymes produce ATP? No, they do not. So the breaking of the bond is not energetically favorable enough for that to occur. Yes, sir.
Could you give like a brief summary of Gibbs free energy?
Sure. Gibbs free energy. So let's go talk about Gibbs free energy. Gibbs free energy is right here. Oh, I do that. Okay, is right here. No, where did I put my? That's the next one. Okay, sorry. It's right here. Okay. Why don't I have? Don't tell me I threw that away. That's glycolysis. So that. There we go. Sorry. Okay. Gibbs free energy. So the Gibbs free energy equation, which I will give you on an exam, says that delta G is equal to delta G0 prime plus RT times natural log of products over reactants. Now, as we look at that equation, remember first of all that the delta G, the very first term, the delta G, tells us the direction of a reaction. Always. Always tells us the direction of a reaction. Delta G is negative, reaction goes forward. Delta G is positive, reaction goes backwards. Delta G equals 0, reaction is at equilibrium. Okay? So, knowing delta G for a reaction, really critical. How do we get delta G? Well, delta G is equal to a constant plus a log term, just like we saw in Henderson-Hasselbalch. Henderson-Hasselbalch said pH was equal to a constant plus a log term. The constant was pKa. The log term was log of salt over acid. Here, the log term is R, the gas constant, times the temperature, times the natural log of products over reactants. And the same rules we had for the signs of the log term for the Henderson-Hasselbalch equation hold here. If products over reactants is greater than 1, this is a positive term. If products over reactants are a value less than 1, this is a negative term. If products over reactants equals 1, this term is equal to 0. Okay? So, to make a reaction go forwards, that's what I was talking about today, to make a reaction go forwards that has a positive delta G0 prime. That aldolase reaction has a very positive delta G reaction. The only way to make the delta G term negative is to make this term negative. And how do I make this term negative? I do it by decreasing products and increasing reactants, because that's going to make this term less than 1. The smaller the number of products and the greater the amount of reactants I have, the more negative this will be, and then that's going to ultimately help this guy to be negative here. It can overcome the positive of this. So, in that aldolase reaction, what was happening? Well, the products were being taken away by that pulling. Pulling was taking away everything after the aldolase reaction. Products were disappearing. Meanwhile, the reactants, which was F16BP, was increasing. This increasing, this decreasing makes this a very negative term, and the reaction goes forward. And that's why pushing and pulling help a reaction to go forwards. Does that make sense? You guys feeling better about this exam than the last exam? What are your thoughts here? No? A lot worse. Oh. You guys didn't feel good about the last one. How could you feel worse than the last one? It's what? Than the last one? Ton of names. Okay, so if you hit a lot of terms and a lot of abbreviations, my advice is always quiz yourself with flashcards. Flashcards are golden for that. Okay? Quiz yourself with flashcards. I think you'll find that to be a very big help. In the process of making flashcards, I always want people to write things down. People make a mistake of saying, oh, it makes sense when I read it. But making sense when you read it doesn't mean that you've learned it. You gotta write it down. It makes a big difference. Other questions? What does the constant domain of an antibody do? It's actually the constant domain. I haven't talked about this, but I'll answer your question. The constant domain of an antibody is what distinguishes the different categories of antibodies. So I mentioned that there were classes of things like IgG, IgA, and so forth, and they have different functions in different places. So you have, for example, I think it's IgE that's commonly found in mucus and tears and so forth, whereas IgG is the most common one that's circulating in your bloodstream. Okay? Those constant regions distinguish the different classes of antibody. That's what they do. Yes, sir.
Is there any other situation like the TRNA enzyme was reading antibiotics goes wrong in the amino acid?
So are there any situations where the tRNA enzyme, the acyl tRNA, the amino acyl tRNA synthetase, that is, would put the wrong amino acid onto a tRNA? The answer is yes. Enzymes make mistakes. DNA polymerase makes mistakes, and amino acyl tRNA synthetase, tRNA enzyme, will make mistakes. Are they common? Fortunately, no.
If that TRNA enzyme did make a mistake, wouldn't that be caught by the EFG?
If the tRNA synthetase enzyme made a mistake, would it be caught by EFG? No, it would not. EFG would have no way of knowing that it made a mistake. Now, what's interesting, I didn't talk about this in class because I figured you guys had enough things to know, so I'll tell you something you don't need to know. And that is this, that the amino acyl tRNA synthetases actually have a type of proofreading in them. They actually have, it's odd and it's hard to describe, which I don't talk about in this class. But they actually have a thing where they flip the amino acid back and forth between two sites to tell if they've got the right amino acid on there or not. And so it actually, if the enzyme discovers that it's got the wrong one, it will cut it off. It's not perfect, like the DNA polymerase was not perfect, but it provides an increased level of integrity for that process.
Is there something that double check when the tRNA brings in that base, when the tRNA, is there something to check that it gives it their name correctly?
So his question, yeah, so you're asking something else here. So his question is there something that makes sure that proper base pairing is occurring between the tRNA anticodon and the codon of the messenger RNA? The answer is yes, but that's not proofreading the amino acid. It takes whatever amino acid is attached there. The protein that does the check to see that the anticodon and codon are properly paired is known as EFTU. That's one of the functions of EFTU. Okay, so basically the question has to do with the wobble codon compared between the messenger RNA and the tRNA. So the messenger RNA has the codon, the tRNA has the anticodon. So if I told you that the third base of the codon was the wobble position, then I could say, well, which position does that correspond to in the anticodon of the tRNA? Well, we remember that they're antiparallel. So the 5' end of one is the 3' end of the other, and vice versa. So it turns out that the wobble base is at the 3' end of the codon of the messenger RNA, which means it has to be at the 5' end of the anticodon of the tRNA. How does what still select the right? It's already been done. It's already been done. Yeah. So what had to happen was the tRNA enzyme had to read the anticodon properly in order to do that. Doesn't matter from that point on. Yep. Oh, that's a good question. In fact, I think I promised to give you guys a structure of ribose. I'm not sure I've done that. So there are, you need to know fructose, glucose, ribose, and sucrose. And I'm going to check tonight. If I haven't put ribose out there, I will. Ribose is very easy to learn. I have? It's on there? Okay. And you're responsible for the linear and straight chain of all those except for sucrose, which only exists in the circular form. Well, I have to ask questions that will rely on your memory of those, right? So here's an example. When I ask it on, so I teach this class in eCampus. eCampus doesn't have anything to draw. eCampus has to answer multiple choice questions. So a question I ask on multiple choice, if they have to know those structures, is, okay, starting with carbon number 1 on glucose in the beta configuration, give me the up-down positions of the hydroxyls, carbons 1 through 4. If we had it in the alpha, down, down, up, down, right? So then they've got several possible answers, and they've got to pick out the right one. So there's, when I said you need to know something, I'm not going to tell you how I'm going to ask it to you. I'm going to ask you in a way that tests your knowledge of it, all right? I want you to use this thing that's in here, right? Yes, sir? Well, each reaction in glycolysis is catalyzed by one enzyme, right? So there's not, you know, from that perspective, they're all important enzymes, right? Yeah. So here's my advice that I would give if you want a strategy for learning these, all right? My advice is learn the names of the molecules first. I'll tell you why. So first two reactions, glucose goes to glucose 6-phosphate. Right? What's glucose? It's a 6-carbon sugar. It's getting a phosphate on there. What's the enzyme that does that? Hexo means 6. Kinase means phosphate. Glucose 6-phosphate goes to fructose 6-phosphate. What's the enzyme that does it? Phosphofructoisomerase. It's isomerizing it, right? Fructose 6-phosphate goes to fructose 1,6-bisphosphate. That's a phosphofructokinase. So if you learn the names first, the enzymes will follow. The only one that doesn't have a clear name is aldolase, and that's the next one. Yeah. Question?
No. I thought you messed up.
Wouldn't be the first time. Yeah. One of the things that you learn when you teach a class is that your mouth can work a lot faster than your brain can. And that's always a problem for me, so. All right, so everybody pretty much got it? Exam is on Wednesday. Cori, C-O-R-I. So there was the Cori cycle. It's an interesting cycle, and it was actually discovered by a husband and wife team back in the 50s. A very rare husband and wife team, with the exception of the Pasteurs. There aren't any other prominent husband and wife teams dating back. Pasteurs were even earlier than that, but dating back into that era. But they discovered that together. They discovered a lot of things relating to glycogen metabolism as well. Yep. Okay.
Okay. In prokaryotes, initiation happens with the Shindle-Garno and messenger formulation.
Mm-hmm.
In eukaryotes, it's just the complex
Mm-hmm.
that helps with the 5' and the polyatomic tail. And it's just that. So this is how you can tell.
Okay. So the question, if I understand it, is the difference between prokaryotic and eukaryotic translation initiation. Okay. So the same process, same general process that happens in prokaryotic initiation happens in eukaryotic initiation. The difference being, number one, there's not a 4-methionine, as you note, okay, in eukaryotes. Okay. You still have initiation factors. You still have all the same things. The difference being that in prokaryotes, they're forming a circular structure, and in eukaryotes, they're forming a circular structure. In prokaryotes, they're not. But they still have initiation factors. The ribosome still has to be assembled. Okay? You don't have a Shindle-Garno in eukaryotes, right? But the differences are fairly minor, actually.
What is the 5' cap and the 3'
So what does the 5' cap
polyA tail do in this?
and the 3' polyA tail do in this? So as you saw in that figure that I drew for you, they are complexed together to make that circular structure. That's all that they do. Okay? So the proteins that are involved in initiation actually recognize those two things and bring them together to make that circle. And that's really all that they do. So, yeah, good question. So does that happen before or after the ribosomal subunits are added? You know, I don't know the answer to that question. I believe that forms before, because that happens pretty early, but I can't tell you that with 100% certainty. You could ask me that on the exam. How about that? So I was talking to somebody in my office today, and one of the guiding principles I'll give to all of you is I know I talk about a lot of things, and I give you a lot of material, but don't make it harder than necessary. Please do focus on the things I talk about. If I don't talk about it, I'm not going to ask you about anything I don't talk about. I can promise you that. I've challenged people over the years. Find something that I've tested you on that I haven't talked about, and I don't think you'll find it. So focus on what I talk about. that hopefully will help reduce the amount of information. Jake.
So the shiny still sequence is on the
Okay.
RNA. It's not.
So where's the Shine-Dalgarno sequence? So the Shine-Dalgarno sequence is found on the messenger RNA. It's complementary to a sequence on the 16S ribosomal RNA. Okay? That's that GGAGG sequence that I talked about. No, you don't need a sequence. See, there's one thing you have to know, right? You guys have it made. I'm making it so easy for this class, right? Okay, I'm sorry to joke about that. I know it's a tough subject, and I've told my classes in the past. You know, I teach BB 450 and 451, where we cover all this material in 2 terms, not in 1 term. So this class, in a lot of ways, is the most challenging class I teach because we go through things so rapidly. And yes, we do go into more depth into things in the BB 450 and 451. But when you're going through things rapidly, it's hard. It takes more time to process and get things into your head. So this actually ends up being harder because of that. This is why I advise my students, okay, who have to take physics, don't do it during the summer. Yet every student wants to say, I want to get it out of the way. And you can't do that. You got to have time to process it. Don't do things like that where you're rushed, because when you're rushed, you're just not going to retain things as well. Okay? My advice for what it's worth. Okay, guys, good luck. I'll be around. I'm gone early in the morning tomorrow, so if you're looking for me early in the morning, I won't be there, but I should be there the rest of the day.